Welcome to Fun Facts Online. Today we're talking about a technology that sounds like science fiction, the ability to edit the very blueprint of life. It's called CRISPR, and it holds the promise to cure genetic diseases, fight cancer, and even revolutionize our food supply. But for all its power, CRISPR has been stuck behind a massive wall. The problem wasn't the tool itself, but getting it to where it needs to go. Now, that wall is starting to crumble, thanks to a tiny, meticulously engineered particle that is set to supercharge CRISPR and unleash its full, world-changing potential. First, a quick refresher on what CRISPR is. Imagine your DNA as a vast library of books containing the instructions for building and running your body. Sometimes, there's a typo in one of those books, leading to a genetic disease. CRISPR, specifically the popular CRISPR-Cas9 system, is like a biological word processor. It has two parts, a guide RNA, which is like a find command that searches the entire library for that specific typo, and a protein called Cas9, which acts like a pair of molecular scissors. Once the guide RNA finds the target, Cas9 cuts the DNA. The cell's own repair crew then rushes in to fix the break. Scientists can hijack this repair process to either disable a faulty gene or, by providing a correct template, actually fix the typo. The potential is staggering. We could theoretically correct the genetic errors behind diseases like sickle cell anemia, cystic fibrosis, and Huntington's disease. But there's always a but. The biggest but for CRISPR has been the delivery problem. The CRISPR-Cas9 system is a large molecule. It can't just waltz into our cells. The cell membrane is an incredibly selective bouncer, and CRISPR isn't on the guest list. Getting this bulky gene-editing machinery into the right cells, and then into the cell's nucleus where the DNA is stored, has been the single greatest hurdle preventing CRISPR therapies from becoming commonplace. Scientists have tried a few different approaches to sneak it past the bouncer, and they fall into two main categories: viral and non-viral. Viral vectors are the Trojan horses of gene therapy. Viruses are, by their nature, absolute experts at injecting their genetic material into cells. So, researchers learned how to strip a virus of its disease-causing parts and replace them with the instructions for building the CRISPR system. It's a clever trick, but it has serious downsides. Our immune system is designed to hunt and destroy viruses, even disarmed ones. This can cause a dangerous immune reaction in the patient. There's also a small but terrifying risk that the virus might randomly insert its DNA into our own, disrupting a critical gene and potentially causing cancer. Finally, many viruses are like compact cars. They just don't have enough trunk space to carry the entire, bulky CRISPR toolkit. To avoid these safety concerns, scientists also developed non-viral methods. These include physical techniques like microinjection, where you use a tiny needle to poke the CRISPR tools directly into a cell. This works fine in a lab dish, but it's obviously not practical for treating a patient with trillions of cells. A much more promising non-viral approach uses nanoparticles, specifically lipid nanoparticles, or LNPs. You've probably heard of these. They are the tiny spheres of fat used to deliver the precious mRNA cargo in the COVID-19 vaccines. LNPs can protect the CRISPR machinery, are less likely to provoke the immune system, and have more cargo space than viruses. However, traditional LNPs aren't perfect. Many of them get trapped and destroyed inside the cell before they can deliver their payload to the nucleus. They can also accumulate in the liver, which can lead to toxicity. This sent researchers on a quest for a smarter, more efficient nanoparticle. The answer came from a revolutionary nanostructure, first invented back in 1996 by a team at Northwestern University led by Chad Merkin. They're called spherical nucleic acids, or SNAs. Imagine a central nanoparticle core, but instead of a simple coating, it's covered in a dense, spiky shell of DNA or RNA strands, all pointing outwards. This unique 3D structure gives SNAs incredible properties. Most importantly, they can enter a huge variety of cells with remarkable ease, without any extra help. The spherical arrangement of DNA allows them to bind to special scavenger receptors on the cell surface, which then actively pull the nanoparticle inside. It's like having a universal VIP pass to get into any cell. This is where the real breakthrough happens. Merck's team had a brilliant idea. What if they could combine the protective, spacious core of a lipid nanoparticle with the cell-penetrating power of a spherical nucleic acid? The result is a new hybrid structure called the lipid nanoparticle spherical nucleic acid, or LNP-SNA. Here's the design. At the center, you have a lipid nanoparticle, a tiny bubble of fat. Stuffed inside is the entire CRISPR-Cas9 toolkit, the Cas9 molecular scissors, the guide RNA to direct it, and even a DNA template for making precise repairs. Then, the outside of this fat bubble is decorated with a dense shell of short DNA strands, turning the entire structure into a super-powered SNA. This elegant design combines the best of both worlds. The LNP core protects the cargo, while the SNA shell acts as a smart coating that supercharges its ability to get into cells. So how does this supercharging actually work? It's a multi-step process. First, the LNP-SNA gets into the cell far more effectively than a standard LNP. While normal LNPs use a fairly standard entry route, the SNA's DNA shell latches onto those scavenger receptors, triggering a more efficient uptake pathway. This results in 2 to 3 times more nanoparticles getting inside the cell. Second, this DNA shell can be programmed. By designing the DNA sequences to recognize specific markers on, say, a cancer cell, the LNP-SNA can act like a guided missile, delivering its payload only to the target tissue and ignoring healthy cells. This is critical for minimizing side effects. Third, the LNP-SNA is an expert at jailbreaking. Once inside the cell, many nanoparticles get trapped in acidic compartments called endosomes, which are basically the cell's recycling bins. But the special lipids in the LNP-SNA's core become positively charged in this acid, causing the nanoparticle to burst out of the endosome, freeing the CRISPR machinery to go to work in the nucleus. And the proof is in the results. In a landmark study, Merck's team tested these LNP-SNAs on various human and animal cells. The results were spectacular. Not only did they enter cells up to 3 times more effectively than the current gold standard LNPs, but they also boosted the rate of gene editing 3-fold. Even more impressively, they tested the system's ability to perform precise gene corrections, a process called homology-directed repair, which is the holy grail of gene editing. The LNP-SNAs improved the success rate of this precise repair by over 60% compared to current methods, achieving efficiencies up to 30% in some cell lines. This is a massive leap forward, proving that the architecture of the delivery vehicle is just as important as the tool it carries. What does this mean for the future? This supercharged delivery system could finally unlock CRISPR's potential to provide one-time cures for a range of genetic diseases. Take sickle cell anemia. The first CRISPR therapy for this disease was recently approved, but it's a complex and expensive process where cells are taken out of the body, edited, and then reinfused. An LNP-SNA system could, in theory, deliver the cure directly into the body, editing the right cells in their natural environment, making the treatment simpler and more accessible. For diseases like cystic fibrosis, where the challenge is getting the therapy into lung cells, the enhanced delivery of LNP-SNAs could be a game changer. For neurological disorders like Huntington's disease, the ability of these nanostructures to cross the formidable blood-brain barrier makes them an incredibly exciting prospect. And the impact isn't limited to medicine. In agriculture, supercharged CRISPR could usher in a new green revolution. Imagine creating crops that are naturally resistant to drought and pests, reducing our reliance on water and chemical pesticides. We could enhance the nutritional value of staple foods like rice and wheat to combat malnutrition, or edit fruits and vegetables to extend their shelf life, drastically cutting down on food waste. An efficient nanoparticle delivery system could accelerate the development of these climate-resilient, more nutritious crops, helping to secure our food supply for a growing global population. Of course, the road ahead still has its challenges. Moving from a lab discovery to an approved therapy is a long and expensive journey. Manufacturing these complex nanostructures at a massive scale with consistent quality is a major engineering hurdle. A spin-out company called Flashpoint Therapeutics is already working to tackle this and move the technology toward clinical trials. And with this immense power comes profound ethical questions. Where do we draw the line between treating disease and enhancing human traits? And how do we ensure that these potentially life-changing therapies, which will likely be very expensive at first, don't create a new form of genetic inequality? These are conversations we need to have as a society. But what's undeniable is that the development of the LNP-SNA represents a pivotal moment. It's a beautiful example of how different fields of science, chemistry, biology, and nanotechnology can come together to solve a monumental problem. The marriage of CRISPR's gene editing power with the precision of nanotechnology has set us on a path toward a future where devastating genetic diseases may no longer be a life sentence. A future where our food is more secure and sustainable. The era of supercharged CRISPR has begun, and with it, a new and exciting chapter in the story of human ingenuity.